Welcome to my channel Biochem Bio World. Today's video is based on the extraction of the DNA, that is one of the nucleic acids from a plant tissue. Okay, and the method which I'll be going to discuss is the CTAB method, which is the most common method used for the extraction of the DNA, and the full form of CTAB is cetyl trimethylammonium bromide. And basically there are three scales. Okay, there are three scales, that is large scale DNA extraction, midi scale DNA extraction, and mini scale DNA extraction. There are only two differences between these three, that is the volume of the solutions added, and the second one is the amount of the tissue. Okay, large scale is usually done in the falcon sausages, and midi is usually done in the Eppendorfs, that is 2 ml, 1.5 ml, whereas mini DNA extraction is done on a tissue lyser. Okay, it's an automatic machine, and the mini DNA extraction is done on that. Well, I'll be discussing large scale and midi in this video in details. Okay, and this is just a normal slide that what you have, the prerequisites, that is the material which is used for the DNA extraction. You need to have 50 ml falcons with you, 25 ml ockridges, it can, I think, up to 30 or 50 maybe, and dispensers for adding the solutions, that is in the large scale. Okay, in the large scale I have to add approximately like up to 15 ml of these solutions, so I need dispensers to make it quick. Okay, and next I need stands for the Eppendorfs or for the ockridges falcons. I need water bath for the incubation. I need a shaker at 37 degrees Celsius. This is one of the steps where we shake the falcons or the Eppendorfs, and I need a centrifugation, that is a cold centrifugation, that is all the centrifugation which is to be performed in DNA extraction is at 4 degrees Celsius. Okay, and 10,000 RPM, and Eppendorfs for midi. Okay, and cylinders, okay, for volumes. If you are not having dispensers, we'll be using cylinders for adding in case of large scale extraction. These are the chemicals which are required in the DNA extraction, that is CTAB buffer, obviously. Okay, CTAB buffer, we need a buffer. Okay, an extraction buffer, and the buffer used is CTAB. I need beta mercaptoethanol. Okay, and it is usually added 0.2% of your CTAB which you are going to use at that moment. And then the third chemical which I require is chloroform is to isomyl alcohol ratio in 24:1. Okay, to crystallize the DNA, I use a chilled isopropanol, and I usually add 0.6 volumes of the supernatant which I have obtained in that step. For the washings, that is 70% ethanol, and last, that is 1x TE buffer. TE, that is tris EDTA. Okay. Next, in the next two slides, I'll be just giving a view of the components of the CTAB buffer, that is NaCl 1 molar tris. The pH is 8, and pH is very important. You have to set the pH prior to using the CTAB buffer. In fact, any buffer in DNA extraction or in molecular work, you have to be precise about the pH of that particular chemical. And 0.5 molar EDTA and CTAB. Okay, I have mentioned the proportions of these components as per in 1 liter. Okay, and the final concentrations as per in that. I told you that we need 0.2% of beta mercaptoethanol. Usually it is not added while the preparation of the buffer. We add when we need to use that buffer, that is during the DNA extraction itself. Okay, and this is the composition of the 1x TE buffer, and the components are TE, obviously tris, and EDTA, and in the concentration of 1 molar and 0.5 molar respectively, and the final concentration as mentioned on the slide itself, that is 10 millimolar and 1 millimolar of tris and EDTA. Okay, and so these are the detailed steps of DNA extraction using CTAB method, and we grind the tissue to fine powder, okay, using liquid nitrogen, and transfer the tissue in case of large scale we use occurages, okay, and in case of the midi scale DNA extraction we use 2 ml microcentrifuge tubes, and I add, after the transference of this tissue, I add a pre-warmed CTAB buffer. The pre-warming of CTAB buffer is usually done at 65 degrees Celsius, and after pre-warming this buffer at 65 degrees Celsius, we usually add 0.2% of the volume of the CTAB buffer, beta mercaptoethanol, and after adding pre-warmed CTAB buffer, okay, into occurages or microcentrifuge tubes, I place these tubes in water bath at 65 degrees Celsius for approximately 60 to 90 minutes, okay, and after this water bath incubation at 65 degrees Celsius, we add 24:1 chloroform isomyl alcohol, okay, and the volumes of CTAB or this chloroform isomyl alcohol is usually 12 to 15 ml in case of large scale DNA extraction, and in case of midi scale DNA extraction it is usually 700 to 800 microliters, okay. After adding the chloroform isomyl alcohol solution, we place these tubes in a shaker for approximately 20 to 30 minutes at 37 degrees Celsius, that is room temperature, and after this shaking step we transfer these we centrifuge these tubes, okay. Centrifugation is done at 4 degrees Celsius, 10,000 rpm, 10 to 15 minutes, and after centrifugation of these tubes we pick the supernatant. In case of the large scale DNA extraction, this supernatant picked is transferred into 50 ml falcons, whereas in case of the midi scale DNA extraction we transfer this supernatant obtained to 1.5 ml appendol, that is microcentrifuge tubes, okay, and whatever the volume of supernatant we have got, we add 0.6 volume of that chilled isopropanol to crystallize our DNA, okay, and we place these tubes for ensuring the complete crystallization. We place these tubes at minus 20 degrees Celsius for approximately half an hour, okay, and after this timing, in case of large scale DNA extraction, in 50 ml falcons you will observe a DNA network, a floating DNA network, okay, and in case of this scale DNA extraction, you pick this DNA network by a blunt tip and transfer it to 1.5 ml microcentrifuge tube. This is in case of the large scale DNA extraction, whereas in case of the midi scale DNA extraction, the solution, that is your the supernatant and the isopropanol solution is already in the 1.5 ml microcentrifuge tubes. As it is, you go to the next step, that is centrifugation. Centrifugation is done 4 degrees Celsius, 10,000 rpm, okay, for approximately 10 to 15 or minutes, and we get a DNA pellet, okay. We discard the supernatant so obtained after the centrifugation, and we give 2 to 3 washings of 70% ethanol, okay, to that pellet. After the 70% washings, we air dry the pellet, okay. We air dry the pellet, and depending upon the size of your DNA pellet, we add the 1x PE buffer to dissolve that DNA, okay. For dissolution, we usually place these tubes at 4 degrees Celsius for overnight for the complete dissolution, and after this we usually check the quality and the quantity of the DNA, either using 0.8% agarose or via nanodrop.